Good morning, everybody. Thank you for joining us today. We'll get started. The Hawaii Coffee Association is pleased to host a series of webinars covering the latest research data concerning the coffee leaf rust disease in Hawaii. We're grateful to all of the scientists whose work is helping our industry to battle rust and other diseases. Coffee leaf rust resistant varietals is a topic of our webinar today, and it's going to be presented by Dr. Tracy Matsumoto. She is a supervisory research horticulturist at the USDA Pacific Basin Agriculture Research Center. Tracy has worked with the Hawaii coffee industry for over 10 years and conducted research on coffee harvest management for CVB control, and is actively involved in establishing the coffee germplasm collection at the repository in Hilo. This webinar will be recorded and published on our website. If you have any questions for Tracy, please feel free to leave them under the Q&A tab, and she will address them at the end of the presentation. At the end of the presentation, I will share the link on our website, so you can rewatch it or share it with anybody after the presentation. Tracy will take it from here.
Okay. Thank you, Ala, and thank you to the HCA for the invitation to present some of our research. And so to give you a quick overview of what I'll be covering today, we're just going to do a quick review of the plant pathology and, what happened? Okay, I'm so sorry. I'm not sure what happened. My computer just shut off, but hopefully everybody can hear me okay?
Yes, we can hear you okay.
Okay.
But the presentation stopped on the overview.
Okay. Yeah. So what we're going to do is I'm going to go do a quick overview of plant pathology and the gene for gene concept. We'll talk about some of the relatively quicker work that we're doing on getting coffee leaf rust varieties, and also looking at the longer term type of solutions by breeding for new varieties for Hawaii. And so just if you remember back from Dr. Lisa Keith's talk earlier, she had talked about the disease triangle. And so this is for a disease to occur, you have to have all three conditions must be met. So you do need a virulent pathogen, in which case ours is Hemilia vestatrix, and you also need a favorable environment. So you need the moisture on the plants, and then you also need a susceptible host. Okay, so to prevent diseases, we can disrupt any one of these conditions. And so through surveying and monitoring, which Dr. Melissa Johnson had talked about, what we can do is we can do field management, such as fungicide sprays. We can do some pruning to increase airflow, also good nutrition for your plants. And so today's talk, what we can also focus on is changing the cultivar of host plant that we're using. And so today I'll be kind of concentrating on this area in disrupting the disease triangle. Okay, so to go back to your, I guess, plant pathology. So back in 1942, Harold Henry Flor had come up with this concept of a gene for gene theory. And so he was actually, this is interesting, I just found out, he was a pathologist in Fargo, North Dakota, and working for the USDA Ag Research Service. And he worked on rust of flax. And what he was able to find was that for each gene that conditions reaction in the host, there is a corresponding gene in the parasite or pathogen that conditions the pathogenicity. And so in the host plant, this is known as the resistance gene, and the corresponding gene in the pathogen would be called the virulence gene, aversion gene. So unfortunately, I had to postpone my talk, but I had some downtime. So I was actually trying to think of a good model to use for the gene for gene concept. And I'm not sure if anybody had ever played the game on your phone or app or anything, and it's called Plants versus Zombies. And so this is actually a nice illustration of the gene for gene concept. And so basically what you have is you have your house, which is your coffee plant, and then you have the zombies, which represent your coffee leaf rust. And so there's a constant battle between the plant trying to build resistance, and you also have the pathogens or zombies trying to attack your plant. Favorable conditions occur, you have your coffee leaf rust. So now, basically what you can do is you can add some plant defenses, as you can see with this border that we put up here. However, if you notice this gene product or zombie with the pole vault, what they're able to do is overcome the resistance. And so this would be like having a susceptible plant, such as our bourbon or typica, which have the virulence genes, which is the host resistance genes, which are either 5 or 2 and 5. However, our virulent pathogen has the 2, 4, and 5 genes. And so that's why we still get coffee leaf rust. And so if we do this where we actually put up increased defenses, like growing a geisha or catamore, what we see is that the disease cannot occur, and basically you do not have diseases occurring. And so those are some of the different methods that we do to constantly, uh, I guess, put up with this battle between hosts. And so if we look at this conceptual in actual reality with coffee, what we can see is that the CIFC in Portugal, who were kind enough to actually characterize our coffee leaf rust strain uh, and note that it was race 24. And so this is an abbreviated list of the differential plants that they have to do. So what they had to do was we sent them the samples, they had to increase the amount, and then they had to pretty much inoculate them on many, many different plants. And from there, by knowing which ones were infected and not infected, they were kind of able to determine which race we have. And so what I did here was kind of highlight the plants or the hosts that are susceptible to race 24. So here you can see the 63/1, which is the bourbon, which is many of the varieties that are grown here. Also want to point out is some of the Timor hybrids, which is that naturally occurring hybrid between the Caniflora and the Arabica. And so you can see that those are somewhat more resistant. Okay. So when we're first looking at, because we know that it's difficult to import different lines into Hawaii, we were looking to see what varieties we might have locally. And so fortunately, we were able to work with some of the growers, and what they noticed in their field that was being defoliated and infected by the coffee leaf rust, they were able to find some trees that are nice and green and healthy. And so what we were able to do is to get some samples from that material and to see what they are. And so at that time, we were fortunate to work with Dr. Daping Zhang, who's an air scientist in Beltsville, and he had developed a set of SNP markers. So a SNP is just a basic DNA change that occurs. And then so by looking at multiple gene or multiple products within the whole genome, we're able to see which ones are similar, which ones are different. And so he was able to use a panel to actually characterize some of the Cartier collection, which is the largest international coffee collection in the world. And from there, from that database, what we're able to do is when we send him samples, he's able to genotype it to see which ones are closest to that gene or closest to that cultivar. So we know in Hawaii that we already have some we well, thanks to his genotyping, we know that we were able to confirm Obata. Fortunate, unfortunate, we were able to also confirm that we do not have 2P, but so it's we like to joke that it's 2P or not 2P, but basically it's the false 2P, I believe is what Mingli is currently calling it. And so, and then we also have a bit of catamores, which you notice in the coffee tree is basically coming off the Robusta branch. And so most of these would be from a lot of the Timor hybrids, such as the catamores and the Sachemores. And so that's kind of mostly the basis of our resistance locally. Okay, so we were able to get some other local variety selections. Here's some of the a resistant variety that Tommy Greenwell had found. And so we know that this is actually similar to 5175, which is actually a catamore. And in the database, it showed up similar to Anna Cafe, but Anna Cafe is actually introgressed catamore line, so which is the Timor hybrid line, the catamore with crossed with a pacamara. And I believe these are resistant to the coffee leaf rust. So he hasn't seen any symptoms on these. And also he was able to cup a few of them and they seem to be cupping very well. And so these are other candidates that would be good for further propagation and multiplication. And so Tommy also had another lines that were selected from Dan Kuhn many, many years ago that were just growing in the fields. And so this one's seem to be maybe out or outcrossed with or open pollinated with some of the other 2P or false 2P and Bourbon. And also, so these were most similar to Costa Rica 95, which is also another catamore background of resistance. Unfortunately, the last I spoke with Tommy, these don't seem to be cupping very well. So these might not be carried on for further testing. Okay. We also worked with some other farmers to look at some of the material early on when coffee leaf first came out to see some that looked a little bit more tolerant or resistant. We did also sample some non-resistant ones at the time. So we do have our natural typicas and everything. But the most ones that are resistant seem to still have that catamore or sachamore within their background, along with some that were crossed with some of the Ethiopian varieties. Okay. So early on, if you had listened to one of my previous talks, we had talked about focused on these three varieties that were shown in that field that I showed in an earlier slide. And so we had sent the samples off to Dao Peng, and he was able to infer that these were Ethiopian and Sarchamore cross, and with the Maite actually having a little bit of Bourbon in it. Upon further study, unfortunately, we found that the cultivars that we called Juan and Cruz seem to be susceptible to coffee leaf rust. Although the infection was a lot lower than some of the typical. So there is a little bit of tolerance. We do see some coffee leaf rust on those. These also have a lower growing growth stature, and they tend to lean a lot more. However, Maite, which seems to have more of a taller growth, maybe it's more similar to the Bourbon, also seems to be coffee leaf rust resistant, and it also cups well. So Tommy was able to get material from this tree and was able to cup it, and it actually cupped quite high. Fortunately, Steve Starnes, who worked with Mike Shintaku on our SCRI grant, had actually initiated many of these lines into tissue culture via somatic embryogenesis. And now what we'll be doing is probably sending at least the Maite to HARC for further multiplication. And so hopefully these will be able to be distributed. Okay, so those are the local varieties which do not have too much restrictions on them. And so prior in, I guess we had started this process with Dr. Chifumi Nagai from HARC in I think 2017, where we were looking at different varieties in anticipation for coffee leaf rust to come in, because we know that importation of coffee is actually prohibited from international sources to Hawaii. And so all coffee must be brought in through a controlled import permit through USDA APHIS. And so even coming from the continental US, we know that the Hawaii Department of Ag has restrictions on bringing in material. So any materials, plants or seeds for propagation will also require a quarantine period. Okay. So in 2017, as I mentioned earlier with Dr. Chifumi Nagai, she was interested in working on the Row Coffee Research International Multi-Location Variety Trial. And so this was established by Row Coffee Research. And so here's a map of the countries. And so if you see cultivar names listed below the country, those are the varieties that were provided by the countries. And then the colored in countries are the ones where the evaluations took place. So roughly they would give you about 30 varieties, and they would grow them in different regions, and they would all take the data the same way just to see which ones perform the best under which conditions. And so I think we were the very last participants in this program because of the whole importation process. And so we finally did get the material in July 2020. However, we did find coffee leaf rust in October 2020. So yeah. But and so these are the varieties that we actually have in tissue culture. So the susceptible versus the resistant or tolerant is based on the Royal Coffee Research Catalog. And so I just want to give you a caveat that although they are listed as susceptible or tolerant, this is in general to coffee leaf rust and not necessarily specific to race 24, which is the race that we have here. So if you notice, if we just looked at the general categorization of these lines, geisha would be considered susceptible to most other coffee leaf rust races around the world. However, for us, it would be considered in the tolerant or resistant category. And so basically, also just to note that most of these cultivars, so when we imported them in, we imported them in for research purposes. And so we signed a contract with World Coffee Research to say that these would not be used for commercial production, but just for evaluation. And so, since coffee leaf rust actually came in 2020, what we wanted to do is kind of look to see if we could get more cultivars for Hawaii for commercial production. And so we had started calling some, and so World Coffee Research had told us that if we get the breeders to sign off to say that we can grow these lines commercially, then we could use the material that was already in Hawaii to then propagate them for commercial production. And so, along with Kimo Faulkner, we started to contact many of these growers. Um, we had many late night calls, depending on what the time difference was, and it was very difficult to negotiate some of these lines. And so, what we wanted to do is kind of focus on which ones we think would have the greatest potential for Hawaii. Luckily, at that time, World Coffee Research had started to publish some of the results that they received from the rest of the countries. And so you can see that they published in general what were the best varieties based on the different criteria. And so here you can see yield, which is one of the more important criteria that they looked at. And here you can see the high scoring varieties are EC16, which is Mundo Maya, H1 Central Americano, Ruru 11, EC15, which is Mundo Mex, and you can see some of Paranima, Marsalesa, IPR 103, and Batian. And so of course, the yield is dependent on the site, which includes the temperature, rainfall, and pest diseases. However, in general, the F1 hybrids, such as the EC16 and Central Americano, perform best globally. And so for disease resistance, we also know that Mundo Maya performs very, very well, even in India, which has many, many different varieties of, I'm sorry, different races of rust. So we know that cupping is important for Hawaii's market. And so this is the WCR global cupping results for 2021, where they use 6 countries, 10 varieties, and 25 cuppers using the SCA protocols. And here you can see some of the top scoring varieties here, which include Marsalesa, Batian, and then EC16, which is the Mundo Maya. So because we were trying to focus on something for the Hawaii market, what we asked World Coffee Research to see is if we could get some of the material for the Hawaii cuppers to test, just because we know that there is a little bit of subjectivity even with some of the tasting. And so we were fortunate to get about 100 grams of green beans. And so because we couldn't import it to Hawaii, what we did is we sent it to a roaster in Tennessee, and she kindly roasted it for us and then sent the roasted beans to us. And so these were cupped locally at Pacific Coffee Research, and a big thank you to Madeline and Brittany for helping us do that. And we were also fortunate that Julie, who's a Q grader from HARC, actually came down for one of our first cuppings. Okay, and so here you can see some of the cupping of the IMLVT lines. And so we had the official cupping at PCR, and you can see some of the cuppers there. And we also did with the leftover samples, we did a unofficial cupping at PBARC, where we had some of our cuppers here, as well as some of the industry folks to come over and kind of test some of the get their thoughts on some of the varieties. And so here, just to summarize, is the cupping of the top. These are the top scoring cups. And so we had a total of 26 samples. We did have that one control Kona coffee. And so these were from 3 different countries, from India, Kenya, and Nicaragua. And so you can tell that just in general, there is definitely a location effect, with the Nicaraguan grown coffees scoring a lot higher. But you can still see some of the trends still apply for some of the genotypes. So this is a great example of your G times E, or your genotype times environment, in producing a phenotype. And so here you have the highest scoring is some of the Catigua, Batian, your Mundo Mayas, Pacamaras. Central Americano actually did well in both Kenya and Nicaragua grown coffee. FLN, Marcelesa seems to score high or low. It seems to be, that one seems to be quite variable. You see our Pacamaras, Mundo Mayas, and your Batian again from Kenya. And so, so from this list, which kind of agrees with what the WCR global cupping had said, we had kind of focused on some of the F1 hybrids and also looking at Batian. But in the meantime, what we had also imported is some of the other material through San Francisco Bay Coffee, and some somatic embryos of Cassiopeia, Centroamericano, Esperanza, Excelencia, and Millennial. And so these are all brought in on a controlled import permit. Unfortunately, the tissue cultures actually have a 2-year quarantine. And so this is still quite a long process. Although we did start these in, I believe, April. So at least time is going by. Okay, so we also are looking, we're fortunate to meet actually Dr. Elijah Charu, who is the director of CALRO, or the Kenya Agriculture and Livestock Research Organization. And so we were able to negotiate with them potential commercial rights to Batian. And so if you notice, Batian was one of the coffees that did score high and did have high for yield and high cupping scores. The nice part about the Batian, it is a true breeding variety of Arabica. So this would be more similar to how you would grow some of the other Typicas, Bourbons, where you could harvest the seeds from your plant and use that to continue to grow on your farm. So it's much more similar to the conventional production, or most of the production that we're used to in Hawaii. And so these are some information that were provided by Dr. Chesarak and Dr. Tichuru. And so the nice part about this is it does have quite high yields, and it is have high resistance to coffee berry disease and coffee leaf rust. Luckily, coffee berry disease is only located in Africa, so but it's something that we don't have to deal with yet. But it's always nice that this would be able to have some resistance here. And so it is a good high yielding coffee, and suitable for different regions in Kenya. Okay, so these are some photos that I just got from a presentation off the web. And so you can see just kind of what the Batian looks like. And so they were looking for SL28 type of growth when they were selecting the Batian. And so Ruru 11 does perform well, and it has high disease resistance. But so the Batian kind of, I guess, captures the best of both. Okay. And so, as you can see, some of our other cultivars are still under a 2-year quarantine. However, I guess at the last HCA meeting, the Hawaii Department of Ag came out with guidelines to import new coffee varieties into Hawaii. And so from here, what we're hoping is that we can work with tissue culture labs in California and Florida to import coffee varieties. And so normally, when we did our old coffee importation, what we did is we brought the plants in tissue culture, and we had to keep it in tissue culture for a year without propagation. And so with this new procedure, what we're hoping is that the quarantine period will be able to be completed part of the time in the tissue culture labs in California and Florida. And so basically, we would just need to bring it in here into Hawaii and do the acclimatization to start getting them, transitioning them from the tissue culture to greenhouse or into the outside environment. And so if that could all occur within the 1-year period, I think it would kind of expedite the quarantine process. And so we're just in the process of working with the labs to get this done. And hopefully this will be another quick method to bring in varieties. Okay, and also we just did receive, so this was just from last week. So we did receive a BSL2 containerized laboratory, and so this container is the intentions of um, actually growing controlled import permits in more soil type of conditions. So far, everything that we've maintained in quarantine has been in tissue culture, but we would like to be able to acclimatize them and kind of get them out to the field a little bit faster. And so this was just set up last week. And so we'll be scheduling an inspection with USDA, APHIS, and Hawaii Department of Ag to try and get this this facility ready for them to certify. In addition, we are planning to build a quarantine facility, which should hopefully be able to house more material to be brought in via quarantine. However, this was one of our quicker methods to try and get things in faster. Okay, and so now we're going to okay, switch gears to breeding new coffee varieties. And so I still have to give a shout out to Dr. Chifumi Nagai, who had the foresight to start doing a lot of this work early on before coffee leaf rust was even a problem in Hawaii. And now her work is being carried on by Dr. Mingli Wong, who's been a wonderful collaborator to work with. And so just to bring up this table again, just remember that what we're trying to do is we're trying to combine more resistance genes into the plant so that we are able to overcome the virulence gene by the pathogens. And this is the reminder for me to ask you to please treat your resistant cultivars with fungicides to prevent new races from forming. And so by looking at the Indian germplasm, Dr. Vareza from the Portugal has emphasized to us quite firmly that you that if you have a lot of resistant cultivars and you do have coffee leaf rust, that you will generate more resistant races. And so even if you can just do that one fungicide or treatment for your cultivars, we don't want to get any more different races, resistant races here. And so for the we have a SCRI project headed by Dr. Mingli Wang that you can see here. And so she is actually breeding some of the resistant Obata and Catamores with some of the high performing varieties in Hawaii, the elite cultivars in Hawaii, like the Tipica, Mochas, yellow Katu'ais. And so the thought process is then this will keep your signature. You could select for your signature cupping qualities, growth qualities, but still incorporating resistance into that. And so this is a nice, wonderful team effort. And so Dr. Lisa Keith, Dr. Lisa Keith's lab does a lot of the screening of the progeny for coffee leaf rust. And also this is being conducted also in HARC. And Dr. Ching Yi Yu will be doing our molecular analysis to identify what the gene products are for resistance, and we'll be evaluating the agronomic characteristics and cupping quality of these material as well. Okay. In addition, we are working with WCR, World Coffee Research, on the Innovia Global Arabica Breeding Network. And so what they did is they got all the data and the material from their IML-VT trials. They selected what some of the best parents were for disease resistance, yield, adaptability to different climates, and they did crosses with that. And what they did is then sent these materials to the different participants throughout the world. And so we all have part of the progeny here. So we do have a progeny from those lines here, and we'll be evaluating those as well. In addition, this photo is nice because there's Dr. Moira Sheehan. And so ARS just got approval to add coffee to the Breeding Insights program. And so what Breeding Insights does is it kind of helps us tie in the molecular analysis with the phenotypic analysis with the hopes of developing a more robust breeding program. So again, with the Innovea breeding program, so these are crosses between parents with different resistances. And I guess the important part here is that all the material developed will be IP free. So any material coming out of this program will be allowed to be used by growers in Hawaii. And so they did have a meeting in Amsterdam where we were able to meet a lot of the researchers there, a lot of the breeders there. And so you can see Dr. Jane Chesarak and Elijah Gisharu in there attended this meeting. And so this facilitated us being able to negotiate the conditions for Batian, which is extremely helpful. And so here you can see some of the Innovea coffee crosses. And so you do see the high performing varieties there and high cupping varieties. And also you can see this SH3 gene. And so I, so in Colombia and some of other areas, they do see breakdown of the resistance to some of the catamores or the Timor hybrids. And so they're looking at different resistant mechanisms. And so SH3, which comes from Liberica, is a new I guess, method that a new source of genetic resistance that they would like to incorporate into the breeding program. And here you can see the Raw Coffee Research Innovia um, members um, taken at their meeting, including some other breeders, Dr. Tanya Humphrey, who is their head of R&D, and Dr. Kraft, who's Asia, Director of Asia and Africa. Okay, so we are working with a lot of different varieties, and so what we and different projects, and so such as the breeding program with Hark. So you can see Mingli and Sayaka. And so the nice part is that we have a lot of collaborators here that we can do disease resistant screenings from Dr. Lisa Keith, and our nematode resistant screenings from Dr. Roxana Myers, and Dr. Ching Yu is doing our molecular evaluation for breeding. And so, I'll be doing the horticulture or agronomic evaluations and multiplication and importation. And so hopefully by working through this pipeline, we hope to give you a good cup of coffee or good product at the end. And like I said, there's so many people to thank. This is definitely a team effort, and we're just so glad to have so many collaborators to help us with this project. It's been amazing that as we started working on this, how helpful everyone that we've reached out to here or throughout the world. They were very sympathetic when we first said, oh no, we have coffee leaf rust, and they've been very, very helpful in helping these projects succeed. And with that, I'll take any questions, and just want to thank and say how fortunate I am to work with such a wonderful group of people that I'm truly appreciative.
Tracy, thank you so much. That
Yes.
was very interesting and educational. I am looking at the Q&A session. I don't see any questions. If anybody has questions, you are welcome to submit them. Oh, one just popped up. So you can go to the Q&A
Okay.
session and
Got it. So how does a rust fungus with no known alternative hosts adapt so well to keep up with the different varieties cultivars that have been introduced? So I think it's just it's uh, uh I guess as the genotypes develop, there is still some recombination within the coffee leaf rust to adapt to the different conditions. And so Dr. Vareza has told us that he pretty much can make up a new race by just transferring it to different cultivars, or just growing it on different varietals. He can I guess asexually create those mutations. Yeah, I don't know, does that answer your question? But okay, also, are there any continuous surveillance of the new races? So oh, I believe Lisa's group is still collecting the leaf races. We haven't, um, other than looking at the genotypes of what they infect. Um, there's no, and we do also are working with the actually the researchers at Purdue University to actually genotype some of them as well. Okay, and Mike's question, what research is being done on the fungus that kills coffee leaf rust? So if you look at Dr. Lisa Keith's presentation, oh, sorry. If you look at Dr. Lisa Keith's presentation, she is working with a lot of mycoparasites, and she is working with Dr. Roberto Barreto in Brazil, and they are looking at different methods to use the microparasites to actually control coffee leaf rust. Okay. Oh, also, is there any plan to market research to understand how the market will accept the hybrids? So I've seen importers have resistance to purchasing hybrids. Yes. So the market. So what we're
Tracy,
hoping,
Tracy,
yes,
you could voice the
read the
question
question.
because yeah, because on the recording later, people won't be able to see it, so
Oh,
read it, that would
gotcha.
be great. Thank you.
Okay. Okay. So the question is there any plan to do market research to understand how the market will accept the hybrids? I've seen importers have resistance to purchasing hybrids thus far, even when they are very proud of donating to the WCR checkoff program. We know producers like hybrids because of the disease resistance and productivity. The sticking point seems to be the marketing for them. So basically, I guess that's why we kind of looked at to see what the markets will look like. The material that we're actually getting in from the tissue culture labs in California and Florida are concentrating on bringing in those uh, F1 hybrids, including Centro Americano and um, Mundo Maya. And so what we're planning to do is kind of bring those in and then growing those in different grower cooperators, and then hoping that they will be able to do some marketing and determining of which cultivars would work best for them. In Hawaii, it's kind of unusual that we do have a lot of farmers that will, or marketing that is directly from the farmers. So we're hoping that giving them the opportunity to actually grow the varieties and develop their own cupping profile, that they will be able to then market it for themselves. And hopefully that answers that question. If not, yeah, please let me know. Okay.
All right, it seems that
I
you've answered
think,
all the questions, Tracy. Thank you very much.
mhm.
I think at this point, we'll close the session. Everybody, once again, thank you very much for joining. Tracy, would you mind if I'll share your email in case people have any questions and they can reach out to
Oh,
you?
yes, no
Okay.
problem.
Yeah, I will put it in the chat session. And we'll close the session for now, and we'll post the recording on our website. Like I said, we have the link posted in the chat session. Once again, thank you everyone for joining. Have a great afternoon. Aloha.
Thank you.